Hi everyone, I'm Sarah, and I'm one of the undergraduate learning assistants for BSci 222. In this video, I'll show you how to work backwards from a genetic or map distance to find your expected frequency of gametes for each genotype. Map distance and genetic distance are interchangeable terms, but what do they signify? The map distance between two genes is calculated from their recombination frequency, and this distance is measured in either centimorgans or map units. Centimorgans and map units are synonymous, so be ready to see either one of these on your exam or your homework. This distance is actually equal to percent recombination, which means that our value for the map distance between two genes is indicative of the percentage of recombinant gametes that you get from crossing over between the two genes. Let's visualize what this means in terms of crossing over between homologous chromosomes. Here's a pair of homologous chromosomes. You have 2 chromatids per chromosome, and the map distance between genes M and D on these chromosomes is 10 centimorgans. We can determine what the parental gametes are by looking at which alleles are on the same chromosome. Okay, so in this case, the dominant alleles big M and big D are together, while the recessive alleles little m and little d are together. This means that the genes are in coupling. Later, we'll look at an example where genes are in repulsion. Now let's determine what the recombinant gametes are. So if we visualize the crossing over occurring, you can draw it out like this. This is an easy way for you to determine what your recombinant gametes are in practice questions by drawing it out to represent the crossing over, and we'll see how we can use this in our second example later. Now this crossing over is what creates the recombinant gametes that we see as a final result of meiosis. As you can see here, there are now 2 parental gametes shown on the top and the bottom chromatid, and 2 recombinant gametes shown on the inner chromatids, right? So the recombinant gametes are seen as the dominant allele big M with the recessive allele little d, and the recessive allele little m with the dominant allele big D on the other chromosome. Here I have drawn the corresponding highlighter color for each recombination based on our first diagram at the top here. So since we're actually given this map distance, we can calculate the values for the corresponding parental and recombinant gametes immediately. So remember that this 10 centimorgans value represents 10% recombination, which means that 10% is the total percent of recombinant gametes. And since there are 2 kinds of recombinant gametes, how do we know what percentage is assigned to each? So you can see there's some math down here, and I'm going to explain this to you. I solved this based on the principle that each recombinant gamete represents roughly half of the total recombinant gametes. This means that we can simply divide our map distance by 2 to get 5% for each recombinant gamete. You may be wondering now, what about the parentals? Where did I get this 90 from? Well, since the recombinant and parental gametes make up the total number of gametes, their frequencies must add up to 100%. So we do 100% minus 10% equals 90% for the parental gametes' total frequency. We then have to divide this by 2 as well, since the same principle of each gamete being roughly half of their total amount still applies. Here's the math for the recombinant and parental percentages. So in this warm-up example, we've solved for the expected percent frequency of each recombinant and parental gamete for genes that are in coupling. As you can see, I made a little summary box here for resultant gametes and the respective percentages for each. Let's take a look at how you might see a question like this on your exam. So this is a kind of question you could easily see on your exam, and it's not going to have any diagrams drawn out for you typically, so I'm going to show you my technique for quickly visualizing the chromosomes on the next slides. For now, let's think about the math here. We're given the map distance of 5 centimorgans, so that is our 5% recombination, and that means that 95% of our gametes will be parental because 100 minus 5 equals 95%. What you should do now is this. Ask yourself whether A and B are in coupling or repulsion. Sometimes the question will tell you directly whether or not the genes are in coupling or repulsion, but in this case you're actually given a way to represent the linkage in the question. In this kind of format, you should look at which alleles are together on either side of this backslash in the middle here. So if you're looking at the top, you can see this represents which alleles are actually on the same chromosome. In this case, the dominant allele A is with the recessive allele B, and on the other side you have your little a, which is recessive A, with dominant B, or big B. Remember in our previous example, the organization of the alleles was actually the exact opposite of this. In this case, we have a small letter with a big letter on each chromosome. This means that genes A and B are in repulsion. So now that we've determined this, let's figure out what our gametes are going to be. So now that we're given, well, okay, so we're not given a diagram, right? But we can actually make our own, and another way to represent linkage that we've learned is using a diagram like this. The line in the middle here is representing the separation of chromosomes. If you can remember from the first slide how I was talking about alleles on separate chromosomes, this line in the middle is representing the separation of those chromosomes, and the alleles on either side are the alleles that are on the same chromosome. And I always draw this diagram out when completing problems like these. And we can now determine what our recombinant gametes are using the same method as before. So here's one recombinant gamete for crossing over, little a with little b. And here is the other one, big A with big B. It's the same idea as when we drew these circles on the actual chromosomes in the first problem. Now let's do the math. So we're going to divide our 5% by 2, and this gives us 2.5%. This means that 2.5% of the gametes are big A with big B, and the other 2.5% are with little a and little b. So this gives us the percentages for our recombinant gametes, and what about our parentals? Let's draw the same diagram here. Remember that our parental gametes are the alleles on the same chromosome. Since this line in the middle, as I said before, is representing the divide between each chromosome, we can see that our parental gametes are the alleles on the same side of the line. So, one gamete is big A with little b, and the other is little a with big B. We know that 95% of our gametes are parentals. So 95 divided in half is 47.5%. This means that 47.5% of the gametes are big A with little b, and the other 47.5% are little a with big B. So to summarize, we went over the different units you can see your map distance in, and what this map distance actually represents. We also use this map distance to calculate the expected percent frequencies of both the parental and recombinant gametes. Remember also that you could use these percentages to actually calculate the numbers of offspring with these gametes. To do this, you would just apply the calculated percentage that you have to a given number of offspring. Just always make sure to read the question carefully and determine whether they're asking for a percentage or frequency or a number of offspring. And finally, we made the really important distinction between what your parental and recombinant gametes will look like depending on whether your genes are in coupling or repulsion. I've written them out here for you for quick reference. I also just wanted to say thank you for watching this video. If you're preparing for an exam coming up, I hope that this helped. I just wanted to remind everyone to take care of yourselves when you're studying for your exams. I have been where you are, and as long as you put in the time and definitely take advantage of all the resources that Dr. Putzel provides, like old practice exams and these videos, for example, you'll definitely do well. Just make sure to get some sleep and stay hydrated and keep on studying, everyone. Good luck!